Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about crystal violet staining. So here I will discuss about the overview of the crystal violet staining and also different application of the crystal violet staining and the protocol. So let's begin. So first is the overview of the crystal violet staining. So on left side, that is the crystal violet structure, and the crystal violet is an antiseptic violet dye that traditionally used to prevent various infections including fungal and bacterial infections. So next is the staining. So crystal violet staining it usually used in the biological experiment to stain the cells. So the crystal violet it ionize to form crystal violet positive and chlorine ion, and then this crystal violet positive it interacts with the negatively charged components like DNA, protein and lipids, and eventually it stain the cells. So you will get the crystal violet stained cells that is looks like the violet color. So next is the applications. So different kind of the staining people that do for a different kind of experiments. So you can check that which one is suitable for your experiment. Then you can do accordingly. So the first one is the staining to check the morphology of the viable cells. So if you want to show people that your very clear view of your cells, then you can do the crystal violet staining. So also the staining co-cultured cells. So here is the two different kind of the cells as you see in the picture. That is one of the cancer cell, that is the bigger cells, and another one is the T cells, that is the round shape small T cell. So when you want to show anything like binding assay or any kind of co-culture assay, then this is pretty much convenient technique to stain the co-cultured cells. So this is pretty much easy experiment, and it's very simple protocol. So here I included the protocol. So first you need to, after culturing the cells, and that is you need to follow the protocol that how many days you need to culture, maybe 48 hours or 72 hours, and after that when you do the staining step, so before that you need to remove the supernatant and then wash the cells with PVS, and then crystal violet solution that need to be prepared in 0.5% 0.5% crystal violet in methanol. So when you use the methanol, then the cell will be fixed at the same time, and you can store it at 4 degrees centigrade for future use. So after adding the crystal violet dye, crystal violet solution, then incubate 20 minutes in room temperature, and after that PBS was 4 to 5 times to fully remove unbound crystal violet from the background. So you need to check under microscope that your unbound crystal violet is fully removed. Then just add PBS and take pictures. So this is very much easy protocol you can follow. So next application, this is the viability and cytotoxicity assay. So here you need to seed around 10,000 cells per well in 96 well plate. So here is the protocol. So here you can follow this protocol. So till this one, that it's all same, that incubate 20 minutes in room temperature and wash 5 times with PBS to fully remove the unbound crystal violet. And then here you need to dry off the plate in dark for 24 hours. So here after adding the crystal violet, then when you wash, and after washing you need to dry off, you need to remove the PVS and you need to dry up the plate in dark for 24 hours, and then for quantification add 100 microliter methanol in each oil. So when you add 100 microliter methanol, then this from the stained cells, the crystal violet will dissolve in the methanol. Then after 10 minutes incubation, measure the OD value. So here the analysis of the cytotoxicity assay, you can follow this this process to get your viable cells percentage or cytotoxicity percentage. So here you should this is the experimental design. So here you need to use one control well that should be untreated. So that means that you can consider that 100% viable cells, that is the highest OD value, and you can see the intensity, it will be the highest in here. And then in these oils, as you see that the viability of your cells, it will be reduced, and as well as the intensity of the color, it will be reduced, and the OD value also, it will be reduced as well. So next you will get the OD value, and then after using that OD value, you can use this formula to get the percentage of the cytotoxicity. So here this is very much convenient, and you can follow this protocol. So next is the colony formation assay. This is also very common technique people use. So here, so this is kind of the cell proliferation assay. So for that, you need to culture the cells in 6-well plate, and usually for the 6-well plate, you need to culture 5,000 cells each well, and incubation of the cells for 10 days, and after that remove the supernatant, PBS wash, and add crystal violet, as I mentioned earlier. The same protocol you need to follow, and then after that you need to take the picture, so you can see that your in the different treatment and the cell proliferation, you can see the different colony formation. So this is all about different kind of the staining technique using the crystal violet. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and if you have any queries, please write in the comment sections. Thanks for watching.